Thank you. So our next speaker is Viola. Welcome.
Thank you very much. Hi, Sharon, and hello, everybody. Can you see my slide? Am I okay?
Yes.
Okay, great. So I have to say I'm very happy and enthusiastic to be here and talk to you today, even though I just realized that I think my talk will be much less scientific as the three talks before, because I decided not to talk about like our detailed research results. Instead, as we are working with mice in the lab, I would like to show why and how can be these mice used in the PXE research. So let me just very briefly introduce myself. So I am Viola. I am from Budapest, Hungary. I graduated here as a biologist in 2005 and had the chance to join the laboratory of András Váradi, who already has been working with PXE. So also my relationship with PXE is started that time. And I even did my PhD work related to ABCC6 and PXE. And I am still currently working here, even though I had a 3-year time period, but I have spent in the US, in Hawaii, which of course my friends and family thinks it was a vacation, but I actually was doing some work as well in the laboratory of Dr. Olivier Lesault. He was one of the three a person who actually found that ABCC6 is the causative gene for PXE. So I had both personally and scientifically a great experience there. But now let's go to the mice. So why, I would like to say why actually mouse is good or is possible to use them as model animals. So first advantage is that they take up very little space. In this photo, I just would like to show our animal facility room, which is only a few square meters, but actually it can host hundreds of mice. They live on these racks in cages, but there is a little close up. So we can put up to 5-6 mice inside a cage. We give them food on the top and drinking water. And actually, when I say they take up little space, I really mean it. This is how they like to be, like 90% of the day being squeezed up together. So it's quite efficient for us space-wise, but also more importantly, they are fairly simple to breed and maintain. There is no special need to keep a colony. Also, their reproduction rate is really fast. The gestation period for mice is only 21 days. And also they grow up really quickly. Just a few photos, that's how a little pup looks like when he or she is 4 days old. After 9 days, they already start to grow hair, and 20 days after birth, they already look like little adults. So we are ready to work with them, use them in the experiments. So, but then most importantly, mice are very similar to humans from many aspects. So we can really model many of our physiological processes in mice. However, I really would like to emphasize that mice are not exactly like humans. So whatever experiments we do, we really have to take into consideration that it cannot be directly translated to human physiology. So what about PXE more closely? There was a group, Arthur Bergen and his co-workers, they generated a mouse strain. So these mice, they managed to delete ABCC6. They used a technique called gene targeting, and I would like to talk about it just really, really briefly. I could talk about it for half an hour, but basically there is a method that you can modify the gene, and you can specifically either add or delete parts of the DNA. And what this group did is they deleted a part of the ABCC6 gene. And with this, they basically disrupted it. And they did this modification in embryonic cells. And then these modified cells were injected to embryos. And in the offspring of after a few generations, they managed to get these so-called knockout mice, where this specific gene, in this case ABCC6, was knocked out, so it wasn't present. It's just a very schematic figure, but actually for this technique, these three gentlemen I show here, they were awarded the Nobel Prize in 2007. So it's a really fancy technique. But so as I mentioned, this group ended up having a mouse strain which had no ABCC6. And these mice showed very similar symptoms than PXE patients have. From their article, I would like to share just one image. Here you can see microscopic images of from different tissues. And you can see that in these mice, for example, in their arteries with this dark staining, you can see they also developed calcification, just like many of you may experience, or in their eyes, in the back of the eyes. Here on the right side, you see a kidney section from a mouse who has ABCC6, but those mice who doesn't have ABCC6, they develop little calcification in the kidney as well. So we learned from them that these ABCC6 deficient mice have this spontaneous calcification, which in human, these symptoms slowly develop maybe in many years. Translating this to mouse terms, it develops in mice in a few months. But then in another group, Zuher Aheraru and his co-workers, they made a very interesting investigations that they realized that certain mouse strains, they also develop calcification after certain injury. We call it like an induced calcification. And in 2007, they managed to show that this calcification was happening in those mice who had no or reduced level of ABCC6. And one way to do such an injury is to actually freeze the cardiac muscle of the mice, and then they develop calcification. So here now you may understand that why it is really useful to have mice, because that's an experiment we surely don't want to do on human. And actually it's not also that drastic on mice, but so I decided to show this one experiment. What we use, these ABCC6 knockout mice are very useful in many ways, but this is one of the experiments I really like, and I would like to show in a few slides what we actually do in the laboratory. So of course we do all these operations under anesthesia, and I also would like to mention here that as in every country, we also have to fulfill very strict requirements. We have to apply for permission to do any kind of experiments on mice. We give them also painkillers if needed, so they are under well control. Okay, but in this experiment, so we prepare the mice for the operation, we shave their belly, and we make a little cut to open the abdominal cavity. And here I just would like to mention that I will show photos like this. So if any of you is like afraid of mice or don't like these figures, please just close your eyes for a minute and I will tell you when to reopen again. So what we do, maybe it's not very visible, but we insert a little plastic tube because we will freeze the heart, but we don't want to freeze any other organs. And for this injury, we use a metal rod, which we put in liquid nitrogen beforehand to make it really cold. And so with this metal stick, for 10 seconds, we press or touch the heart of the mouse through the diaphragm. And then after that, we basically put sutures in the muscle and in the skin. And when the mice wake up, they are really fine and happy. And in contrast to the spontaneous calcification, which happens in many months, we have to wait only a few days to get the results. And now I will show photos of the dissection. So if you don't like that, please close your eyes again. So we open up the mouse. So this is the abdominal cavity. You can see the liver here. That very thin layer was the diaphragm. We put the metal rod right here and touched the heart. And when we open up the chest, here you can see the lung, and this is the heart. And here maybe you can see better here on the heart. This little round area here was the site of the injury, and that white ring around the injury site, that's actually calcium calcification. So end of horrific pictures of mice. So to determine the amount of calcification, we use one of the two methods. One is a colorimetric assay. Here we use an assay mix. It contains a molecule which, after adding calcium, it changes its color. The more calcium we add, the deeper purple the color will be. And luckily, it's very easy to, using a photometer, to quantify. So we actually can get a number. What we do, we usually make a nice calibration like this. adding known amount of calcium to our solution. And this represents known amount of calcium. And then we measure our samples taken from the heart. And the more calcium the heart has, the deeper color we will get. So we actually will get a number at the end. The other way is to use histology. Here I show an example of the heart calcification. You can see cross sections of mouse heart. Here, this is a, let's say, healthy mouse who has ABCC6 in both alleles, while here you see the one, the knockout mice who doesn't have ABCC6. And you can see here sites of calcification after a certain staining. So this is the background. So I show this technique because we really like to use this. Because as I said, with the colorimetric assay, we can do really precise calculations. And let me show only one example from our research studies. When we do this somewhat dramatic cardiac injury to a healthy mouse with ABCC6 gene and protein, there is very low amount of calcification, barely no calcification at all. But as I showed in the previous slide, in mice who doesn't have ABCC6, they develop high calcification. And this we can use as the negative and positive end of a scale. And what we are doing is, of course, trying to cure the phenotype, trying to get rid of the calcification or prevent calcification. And if we, in parallel to this freeze injury, if we are treating the mice with whatever compound we want to test, we expect that the results will be somewhere in between these two data points. So for example, recently pyrophosphate became in the focus or one of the focus of research, as you could also hear from Wilco in the previous talk, that pyrophosphate and its analog bisphosphonates may be efficient. We also tested that ABCC6 knockout mice were given pyrophosphate while we were doing the surgery. And we were very happy to see that indeed the pyrophosphate treatment could prevent the development of calcification. So we are very enthusiastic at this point, but again, as the previously you heard, there is a long way to go until it can get to human practice. There are many things to be clarified. Just, for example, what kind of pyrophosphate to use, because it exists as a salt. So there are other ions connected to it. Sodium, for example, and too much sodium is not good. So we are right now testing different salt forms. The dose is not obvious. The formulation, whether it's taken in capsules, what kind of capsules. So there are many questions to be answered, and it's absolutely necessary to perform well-controlled clinical trials before we can say that it's safe and it's efficient to use. So just again, as much as we would like to translate it directly that, yay, it works in mice, let's take it. Unfortunately, it doesn't work that easily, but we are very optimistic, and that's what we do to work on it to make it happen. So with this, that was my little story about mice. So I would like to thank you for your attention. And here just would like to mention that, so as I said, I work in the laboratory of András Váradi, and currently it's a fairly small group. We are doing these experiments with Esther, Nati, and Kristi, and with the help of great assistants, György is helping to prepare for us all the experimental material in the lab, and Gabi is taking care of our cute mice. So I am happy to answer any questions also right now, or if you will have questions later, here is my email address. Please feel free to contact me. And again, thank you for your attention.
Thank you very much, Viola. Very good. Thank you actually for going through that, because it's something that we think we jump over and don't always give much explanation of, and that really helps a lot. Questions? You can either raise your hand or put them in the chat. Or some thank yous. Okay, good. Thank you. That was such
really, I am in the future also happy to answer questions if it will come up later. And thank you, Sharon, for organizing this session. I'm really enjoying this. Thank you.
Great, wonderful. Me too. It's going very well. Thank you.